introduction. Everybody knows Dr. Lucia Cevidanes. Lucia is faculty at Department of Orthodontics and also professor of dentistry. She is one of the few faculty in the School of Dentistry that has been collaborating with our department. And I would say perhaps one of the few faculties at national, at nation who's doing this much 3D image processing and has integrated advanced machine learning and image processing technique into the clinical decision support systems. She's been supervising a number of our students, co-supervising a number of students and postdocs in our department, research faculty. She has over 140 scientific papers, most of them on data science and 3D imaging applied to dentistry. She has won so many international awards, and she's currently conducting multiple funded research that are funded by different agencies, including National Institute of Health. She's been a great collaborator of our lab. So some of the people in our lab have been co-supervised by Lucia, and we are very happy to have Lucia here as our speaker for this week. So without any further delay, Lucia, we're very honored and happy to have you with us this week.
Thank you so very much. I am the one who's honored. So let me share my screen. And hopefully over the next half hour, I will share with you the work that we have been honored to collaborate with researchers in your department on applications of decision support systems in dentistry. Our work that is currently funded, we received in R1 to apply data science approaches to investigate temporomandibular joint disorders that affect more than 10 million Americans. And this year in 2020, we have been fortunate to have received also an administrative supplement for Reza Surushmer that has worked very closely with our lab with Kevin's guidance since 2019. And our work really fits into the NIDCR 2030 goal areas of supporting research toward precision systems in dentistry, as well as they do have a new portfolio and new funding announcements that we are looking very closely for our new applications on decision support systems and data science approaches applied to dentistry conditions. The joint that we study is this very small joint here that we all have to use our mouths to chew, but it has very different characteristics than the larger joints that get the bulk of the research money, because these conditions, for example, of osteoarthritis in the knee, it's very debilitating, while TMJ, people most of the times they just live with this condition, but these situations can also be extremely painful that lead to total joint prosthesis. But in some cases, these conditions start in an asymptomatic way, and by the time that the patient is diagnosed, so much bone destruction has already occurred, and that's when total prosthesis is needed. And our work has taken an approach of looking at the etiology of arthritis, not just trying to identify one single marker that would be the driving factor in this condition, because we know there is a crosstalk in the molecular pathways of inflammation, angiogenesis, neuroception, bone apposition, and oral resorption to allow us to really understand how osteoarthritis develops. So the proposed work under our funded R1 is started mainly with shape analysis, and this is just to show like how the condyle in the lower jaw looks, in patients that do not have the disease, and how some bone destruction starts to occur initially with proliferation of bone in the anterior aspect of the joint. But it's in long term, the disease is degenerative, with the condyles becoming a lot smaller and a typical bone destruction in the lateral portion of the joint. In our previous work, in 2018, we had identified some different groups and stages of the degeneration, but looking mostly at the 3D morphology and at early diagnosis. Even in stage 1 of the degeneration, we had already seen marked bone loss. We also did, since 2014, in the work that some work that I started at UNC, some statistical multivariate functional analysis to correlate the level of the markers with specific areas of the condyle. This work, I think, was very interesting, but we did get some criticism in the sense that how is it possible that the level of the markers will really correlate with the meshes and the thousands of points, because this is a high dimensional problem, and how robust are these correlations. So moving on to the aims for our current work, utilizing data science approaches to identify biomarkers for classify the disease. We have here at U of M since 2014 and 15, initially funded by some pilot projects from foundations and from MISHAR too, and from the early stages of our funding, because we are now funded for years 6 to 10 of this project. We developed a protocol to improve the quality of the images that we acquired. They are cone beam CT images. They are not as high resolution as the CT scans that you take in the medical field, but they are better qualities than we started with in 2013 and 14, as well as added very detailed protocol for acquiring biological samples from blood and saliva, and a standardized protocol for clinical exams and surveys as part of all of the patient demographic information. All of this data is stored in web-based data managed system that our research team has developed, that is called DSCI, Data Storage for Computation and Integration. And this is just the login page of our DSCI that is responsible for not just storing, but computing different tasks. And Kayvon's group has already developed a couple of tools that have been implemented in this web system for different centers in the US, not just for Michigan. And the goal of the different tasks do allow us to model not just the imaging data, but the biological and the clinical data too, as well as here just saying it run the quantitative data that is computed from the saliva and the plasma, the very detailed information from the clinical evaluation, pain variables, mouth opening and demographics. And from the CBCTs, we now not just utilize the 3D surface, but also quantitative radiomics and both bone morphometry. The first work that we did with VRESA was to do some image processing to extract the bone features added to our previous work on 3D shape analysis. And we had identified initially 17 biomarkers that were expressed in TMJ OA patients that were cytokines, chemokines, growth factors, and neurotransmitters, as well as apoptotic regulators and enzymes and coagulation factors in blood and in synovial fluid. We have since then stopped collecting synovial fluid because it's more invasive for these very small joints, but have demonstrated that these proteins are also expressed in systemic fluids such as saliva and plasma. And this is the work that was published in 2019, then looking at the detailed information of how the texture of the bone, the quantitative information changes in the disease compared to the control patients. These are some of the CBCT large field of view that are acquired regularly for most of our surgical patients. And this is the protocol that we have been utilizing. When I talk to Keivan, I say, these are high resolution images, and Keivan says, wow, these images are so noisy. These are bad scans. Don't you have anything better? And I said, no, we don't have the money, or we can't irradiate the patients because CT scan is a lot more radiation dose for these patients. These are low dose imaging systems that were originally developed for radiation therapy, but they are a lot less cost for the patients too. From then, we have moved to analyze this data. We have implemented some machine learning approaches to analyze the data that we have collected prospectively. Some of these machine learning approaches have not yet been implemented in our web system, but we will be continuing to work with our research team towards implementing different machine learning approaches into the web system, including in the supplement that we just got. We are fortunate to have Reza working with a postdoctoral fellow in Keivan's lab, Emmanuel Zapala, on adding privileged information in machine learning approaches to disease classification and prediction of disease progression. This is our work that, in fact, now it's not under review anymore. It has been published in scientific reports now in 2020, where the two deep learning approaches utilized were XGBoost and LightGBM. First, our statistical expert from UNC utilized a general association analysis of risk factors to select not only biomarkers, but interactions between the biomarkers that would properly classify the disease condition. And we have moved in from there to compare, because all of the image analysis require quite some image processing. We have been working with Reza to combine active deep learning and active contour modeling. The tool for automatic segmentation of the condyles has already been implemented in our web system, and clinicians such as our PhD student in the lab, Najla, are being able to utilize this tool to actually segment automatically all of the follow-up condyles, because until the beginning of this year, we had analyzed only our baseline data sets, but we are now collecting 2 to 3 year follow-up for changes in assessment of changes in long term. And in the methods also developed in Keivan's lab, Reza is working with Emmanuel to apply game theory for feature selection and compare this with some of the other methods published in the literature and that our group had already implemented. So we are very fortunate to have added new steps in our original proposal, thanks with these very exciting new collaborations with the Michigan Institute for Data Science. That has made a big difference for us. And of course, the goal is that once these models are deployed, and provide new insight into this condition and new decision-making support. Our studies are observational. They are not any intervention, but we hope that this will give us targets for future treatment personalization. In identifying the biomarkers, we have up to now, as I said, utilized mainly as procedures like GBM and XGBoost. Emmanuel is working with Rez in both SVM and SVM Plus, utilizing privileged information to see the performance of those approaches. And we had discussed with Keivan also utilizing in long-term for the follow-up assessments, non-convex kernel learning, and will follow their guidance. So with our goal to improving decision-making for the TMJ osteoarthritis diagnosis. And this is just a diagram, and I'm not going to go into detail because it's too much information, of our predictive model of TAJ-OA progression, as we are also collecting 2-year follow-up data. The baseline visit, we acquired both imaging, clinical, and biological markers, and these are some example of surrogate markers in our preliminary studies, and we are now collecting the longitudinal data that we have not analyzed yet. So we're very excited to continue this work. That's the part of our work that has been funded. We, I think, have been very fortunate to have gotten foundation support, NIH support. That's just one R1. But our recent work, we are actually gathering some ideas and towards what we call a patient-specific classification of dental root canal and crown shape. We're aiming to get this ready for a submission for March in this new initiative on decision support systems in dentistry. And the idea is that and most of you have probably seen in your Instagram, your do-it-yourself aligners. You can send them just a picture that you take on your cell phone, and they move your teeth with aligners. But those are based just either on reconstructions from only the crowns with no information of where the roots of the teeth are. So this is in a specific application in the field of orthodontics, but the importance of integrating information from the crowns with the root canals, it extends to many different areas in dentistry, such as endodontics, implant dentistry, restorative, because the crown of the shape needs to match of where the root is. And so this requires combining different imaging modalities informations that include low contrast of very narrow and tortuous root canals with the greater anatomic detail of the crown morphology obtained by intraoral scanners. And here we're clipping the gums here because our goal is to have the whole tooth information from the digital intraoral, and we have done some preliminary work for registration of the information from the volumetric images to the surface scanner images. Our preliminary work with Kevan's group and Reza leading this work has utilized first image pre-processing for automatic segmentation of the root canal, then deep learning with UNet. And this work actually is a summary of, it was accepted for presentation of the Shape My workshop in the beginning of October. It's a MICAI workshop. And then in the end, we do need to do a little bit of image post-processing. So this code was developed by one of our previous French interns that worked under Reza's supervision. And we're looking forward to having new interns with all of their DS 2019s were just approved, so they will be here in the beginning of October to hopefully implement the code for this work also in our web system for clinical applications. And we have integrated this work that Reza did with our collaborators in computer sciences at UNC that implemented the tool for automatic segmentation of the dental crowns from those surface scanners that are very easily acquired in private practice now, and all patients take the digital scanners that utilize some projection planes to probe the digital model surface and extract associated shape features that separates the gums and the teeth. And this is the architecture of the RUNET neural network that our colleague from UNC has implemented. This part of the pipeline of this tool has already been deployed in the web system, but will require further refinements in pre-processing, post-processing steps, as well as identification of each tooth separately and merging to the root canal information. A summary of our results in this, in the CBCT images. This is a manual segmentation by experts, even if it's time consuming. Clinical experts with any of the tools that we use for segmentation often miss the apical portion of the root canal. This is rendering of the manual segmentation. the segmentation with using only image processing, and here combining image processing and deep learning approaches in the tool that Reza has developed recently with a French intern, Mr. Maxime Dumont, that some of you in Kayvon's lab may have interacted with. Our preliminary results in terms of testing area under the curve, sensitivity and specificity, and the segmentation of the crowns and identification of individual tooth morphology with the respective confusion matrix to detect the accuracy of this precision. The ultimate goal of these pilot studies is then to combine information of different modalities towards determining the position of the root inside the bone. And this will hopefully lead to avoiding undesirable tooth movement or undesirable placement of implants inside the bone, as well as guide tools for endodontic treatment. And in just a diagram here of why this is relevant, up to now we have had from the digital scanners just information from the crown, and because of the quality of these images, it's very hard to see where the root canals are. But of course, we can't forget that these teeth are inside bones with very little bone coverage, so the precision of the treatment needs to be taken into account. Otherwise, as a result of, for example, tooth movement with aligners, the roots of the teeth can be thrown out of the bone with tooth loss in the long term because of inadequate position. Our work has only been possible because of the truly interdisciplinary nature of the work. These are some of the people from the dental school and some of the people that you recognize in your own department. And we have been very fortunate to have had some mentoring of Dr. Roman and Dr. Athey, for which we are very grateful for. I don't know if I sped up and talked too fast, but that's what I had prepared to share with you this afternoon. Thank you so much for listening, and I'm ready to hear feedback and comments for the challenges that we have to face ahead.
Thank you, Lucia. So I think it's sort of a record of fitting so many different systems, a schematic diagram results in a presentation in half an hour. That was like a breakthrough. So thank you very much, Lucia. So any questions for Lucia? If you have a question, please activate your, unmute yourself and ask the question.
Yeah, hi, Lucia. Hi, that was great. Thank you so much. I'm glad to see all the progress. You know, I was just curious about the timeline on the bone pathology and the TMJ. You showed that slide where you had the normal ones and then kind of the ones that were progressing and then the ones that were, you know, really kind of stuffed out. You know, how long does that take, or does that just vary with the severity of it? And is there any kind of, you know, kind of any kind of deeper understanding that from your experience that you've been able to get in terms of, you know, and then also when you know, do you get the locked jaw and stuff when those bones kind of deteriorate like that? Does that make it just keep getting worse?
I was actually trying to go back, and right now I don't know if you guys can see that my computer is frozen.
Oh no!
Can you see the little thing twisting here? That my computer is frozen, or you can't see that in the video recording?
No, we can't see that. It looks fine to us. We see the cursor move.
I stopped sharing, but yeah, it's my PowerPoint that is frozen. I stopped sharing for a little bit. Oh, but now my PowerPoint's frozen. You can still see me, right? Because
yes.
Yeah, we can see you. Everybody. So stop and then try sharing again. That's what I did.
Well, I can't, because once I clicked on stop sharing, it went back to my PowerPoint screen, and my PowerPoint screen is frozen, and I can't do anything.
Well, you can just answer in words, though.
Sure. Yes. With regard to disease progression, It is true that some patients have a lot faster progression, and this is one of the reasons that we are looking at such detailed markers, both at imaging level, biological level, and clinical level, because we do not know the answer why some patients have such more rapid bone destruction, while others will go decades without having marked bone loss. But there are the patients with acute stages of the condition where they have the joint that is mainly completely flattened and resorbed in about 1 to 2 years. So in our estimated follow-up of 2 years, we actually have a couple of very interesting cases that have already come back, where the joint was completely remodeled during the observation period.
Oh, wow.
Yeah, so all patients actually had some only conservative approach. We know that there is a retention rate when you're doing any longitudinal observation study, that some of the patients didn't return because they actually had surgery done during the observation treatment, which is a confounding factor, because maybe some of the more severe patients, we actually lost them because they decided to be treated surgically during the follow-up period. But even then, we're documenting the patients who sought surgical intervention during the treatment, as well with the patients that had rapid disease progression. The challenge with that, and as a follow-up, as I mentioned, we are fortunate to be funded for years 6 to 10, and it was the work that Kayvon and the feedback that Gil and you, Brian, did that allow us to renew this project for 5 more years. So this would not have happened without your input. But moving forward, our goal was also for the TMJ project to develop UO1 with different clinical centers, and we have started, I didn't add that information in this presentation, we have started some preliminary work with the Oregon Health Sciences, because they have recently gotten an R1 on longitudinal changes of the TMJ, but their data has been, so hard to work with, because it's like the large field of view scans that have very poor image quality. They have both CBCT and they have MRIs, but their MRIs were not acquired with a 3-dimensional imaging protocol. We see all of the MRIs in the medical field, even MRI elastography that even detects the muscles and so on. But those are very high quality images that are usually not taken for the TMJ. The MRIs of the TMJs are taken with very thick slices that do not make 3D reconstructions possible, and doing the multimodality integration has been a challenge for the data that we have gotten from different centers. So those are challenges for the future. How do we integrate data from different quality of imaging? Because for these approaches to be successful and really implement deep learning approaches, we need large number of data. But every large data set that I have looked at really suffers from the quality of the data set, from the standardization of the protocol in which the data were acquired.
So I have a question for you.
Yes.
I'd like to know the extent to which your PhD colleagues and dental colleagues, besides participating and appreciating the analyses from Kayvan's team and from you yourself, extent to which they are learning. Is that a goal, and how is that coming along?
Gil, I don't know if you were cut just for me.
Yeah, we didn't hear part of that.
Yeah.
Repeat like the last half.
Gil, are you there?
Yeah, I'm here. Is this better?
Yeah, we just didn't hear you.
Sorry. I was asking about your PhD colleagues or trainees and your dental colleagues and trainees, whether they are contributing data and using your analyses, or whether they're also interested and capable of learning to do machine learning so that they actually can take over some of these projects and not just be dependent upon Kvan and you.
No, yeah. So the goal of the web system that I shared briefly is to have the tools as clinician friendly and as automated as possible so that clinician can apply these tools. That's our goal. We don't have a large number of users outside of our group at U of M now. But we are training people with that regard and how to use the web system, and it's still in the development stages. Juan Carlos Prieto, our collaborator at UNC, is the one who helped us with the implementation of the web system. But just for you to have an idea, there are many initiatives outside of our group, and there are 2 different major journals in my field that are doing special issues only on machine learning applications to dentistry.
Yeah. Okay, well, that's a little different from my question.
Yeah, but I mean, are the tools readily available for the clinicians to use and to apply? Is that your question, right?
No, I understand you're setting it up so they can apply without understanding anything about how it works. But yeah. I'm curious whether any of them are trying to learn how actually to create machine learning algorithms and to make assessments of them or modifications of them. Now, are they trying to get behind the setup that you've already prepared for them just to be users?
Yeah, I would think that clinicians can only be users, honestly, because
that's what we say about other fields too, but it's not true. It's just that not most of them or have the interest or the background to do the laboratory work or the mathematical modeling or such. But that doesn't mean that a few of them might not have the capability to actually learn if given the encouragement. That's why I'm asking.
Yeah, even myself, I could not do the work without the developers that I work with. I do not have enough understanding of the mathematical approaches or the programming approaches that it takes. And we were very fortunate to have a postdoctoral fellow with us for 2 years that led some of the work that we have done, and he is now being recruited as a faculty at the University of Pacific that we hope to continue to be one of the centers that we collaborate with. But even somebody with Jonas that figure out some of the statistical approaches, he doesn't have the understanding to actually implement the machine learning approaches. And from what and Kvan can please correct me, or I don't know if Reza or Emmanuel had a chance to join this presentation, but for each application, it requires so much programming, like the machine learning approaches are not generalizable. For example, if we have one of the biggest issues that I was describing is that the machine learning approach for it works very well for segmenting the condyles, but when we started using images from different centers, it was much harder to make the machine learning, the deep learning approaches, even combined with image processing, to work properly. And that's a tool that Reza still needs to develop further with our next programmer that will join. This programmer worked through COVID remotely until the end of June. But after he left, we haven't made any progress on the code for automatic segmentation of images from different clinical centers. So, and if, Reza, are you here with us?
I didn't see him.
Okay, so yeah, Kvan, maybe you can talk with that regard, but for example, the postdoctoral fellow, Emmanuel, who's working with Reza to implementing privilege information in machine learning approach, he's currently working only with SVM and SVM+, and Reza tells me that it will take him a couple of months to be able to test other machine learning approaches, even at a postdoctoral fellow level of expertise in your field. So this is why I'm saying I don't think that clinicians lack the interest, but at least in the field of dentistry, they are looking for algorithms that can be applied for the actual problems in dentistry and applied in a clinician-friendly way. Maybe clinicians are too dumb to understand how we do, but from my understanding on this, and Keivan, please correct me if I'm wrong, the machine learning approaches are not generalizable for different applications or different types of images, and they need to be carefully, it takes quite some time and work to develop the proper algorithms. Do you want to add to that, Keivan?
So Gil, I think you're asking a very good question. And I would say the compromise, the sweet spot between, like, clinicians sitting and writing the code and, testing machine learning algorithms versus the part that they only have people who are doing that and they stay aside. The sweet spot for that is somewhere in the middle. And Midas has actually started, one of the programs that we just started with Midas is the data science training programs for clinicians. So this program, it has like, a workshop that we talk to people, but then it has like a 1 week of boot camp that we bring clinicians who may not have any background in computational sciences. And what we cover them, we introduce different types of machine learning algorithm and some mathematical background that they need to understand what machine learning is doing, right? So and that would enable the clinicians to understand all the machine learning, the general concept, and participate in the decision making as to what algorithm to be used and how to test it. So that's a different thing that clinicians, we teach them some programming, but we go over major methodologies in machine learning, helping them be more on top of the problem. Again, in the end, we are telling the clinicians that this training does not give you the ability to necessarily sit and code your own algorithm or design the mathematics for a new mathematics.
I understand. Coding is another level of detail altogether.
Right.
But strategizing, even having an intelligent conversation would be a nice
And the
step.
School of Dentistry, Gil, has been involved in it. School of Dentistry has a representative in the curriculum committee that designed the curriculum for this boot camp, and we are recruiting. Now we have maybe that might be like a good opportunity to advertise for that, Gil. If you and Brian can advertise it across medical school, we want to, and dental school, we can bring like clinicians and give them what they need in order to be more involved in the design.
Yeah, well, send something. We can help with that, sure.
Yeah, that's good. That'd be very good.
That's very good. Thank you for
Thanks.
that.
You
see
can
Reza showed up, Lucia.
Yeah, you can prepare it, Keivan, and just show us, and you can carry it forward. You don't need us to draft something up for you on this subject.
Oh, we have the curriculum. We have the flyer.
Got that part.
Dates.
We're just gonna help them send it around to our friends.
Yeah. I'll send the flyer to you guys soon.
Okay. Thank you both.
Sorry for my sound. I still have
Boy, Lucia, I know you're having some trouble now with the audio.
Yeah, I'm actually hoping you hear me better now.
But we do have Reza on the line now. So Reza, I'm glad you showed up. So do you have any, anything you could tell us about, your work with Lucia from the deep learning point of view?
Hi everybody, sorry for being late. So I had another meeting. So currently we are working on, I don't know if Lucia mentioned or not, on the privileged learning. And we're gonna use molecular data as privilege information. And we are working on developing a deep learning model that accepts privilege information. So currently we have SVM Plus that accepts privilege information, but I guess the concept of using privilege information in deep learning is something very new, and we are working on it.
Yeah, so I'm involved with Keivan in another project with Nick, so I've got some kind of idea of the privileged information. So maybe Keivan can say a few words about that, just how that works.
So basically, standard machine learning has primary information that makes a prediction about the output, the class, the diagnosis. So if you have primary information, you have a mapping. Deep learning, the machine learning is a function that maps primary information to the outcome. There are cases that besides the primary information, you may or may not have access to secondary information, let's call it privileged information, that may help with the process of decision making. But keep in mind, we still want to map primary to the outcome. But can we, in the training process, can we let this secondary information, privileged information, help us with the mapping and create a better mapping between primary information and the outcome? For that, I give the example that Reza said. So in majority of the cases, in the clinical setting, we have the images, and based on the images, we want to make a decision that, like, you know, a dental problem is there or not, like, you know, TMJ has some issues or not. But we have only X-ray images. It's very unlikely that in clinical setting, you will have access to molecular data. Nobody is running any kind of molecular assay at the clinical setting in most cases. But in the research level kind of, you know, work, we have both molecular data and the imaging data. So the question is, can we use the research data that has all of that and still map imaging data to the outcome, but use molecular information as privileged information that help improve the mapping between primary information, which is imaging, and the outcome? That's what Reza is working, and we're trying to apply privileged information on multiple, you know, standard and new machine learning. What we're doing with Brian is we have applied privileged information to SVM. We are currently, and that is what we are now applying to the application that Lucia has presented to us. But we are also working on different types of regression trees, random forest, neural networks, and naive-based models, and we're creating the version of privileged learning version of all these standard machine learning together with some of the new machine learning that we have created, and Lucia was referring to flexible, non-convex kernels that we are working on.
I was really curious about, you had a couple words on your slides, active deep learning, and there were some other keywords that were active. Can you say a little bit about that? What does that mean?
Reza, you can help us here, but basically majority of the cases that we don't have enough sufficient data to rely on a passive approach to actually train the model, right? We don't have enough examples. With active learning, whether it's applied to deep learning or other methods, we are at any given point, we look at the data that we have and we ask the question, what would be the next most informative example that can help us train the model better? And in those cases, we actively search for such a case, and we seek, like, you know, data from patients who have this ability, and it helps us to have a faster convergence in the learning.
Right.
So most of the active learning versus passive learning, the performance goes like that, and active learning goes like that. With much fewer examples, we can achieve the same level of accuracy. But Reza, we are applying that currently to different types of machine learning. Can you give us an update on that, if you have results or?
Not for the TMJ project currently. So for the TMJ currently, we are working on just incorporating the privileged information to SVM and to other networks, as you mentioned, like deep learning models and neural network models. But
Reza,
for other projects, yes, yeah.
Rez, I don't know if you used active learning already, but how about the work that you did with Maxime Dumont that first utilized or even with search for the segmentation of the scans from the different centers. And this is what I was mentioning to them until my computer froze and I will need to restart it. So I'm using my cell phone to talk because it was causing a lot of echo here. The work that you did with Serge Brosset, and the code still need to be further developed. He did utilize first image processing, which is active image processing, and then he utilized a UNet to train the models for automatic segmentation of the region of the TMJ and the mandibular ramus, right? Was that not active learning yet?
Yeah, so the one that you are referring to was using the active contour as a method of image segmentation. So we used active contour.
No, I'm talking about the methods that he hasn't deployed yet, not the imaging processing work that has been deployed.
Yeah,
but the one.
Lucia, we have
Yes.
the mathematics, we have worked on the mathematics, but based on what Reza is telling us, we haven't actually yet applied that. We use that for other applications, active learning, but for this application, apparently we haven't yet used it.
Okay. Well, so we have more work to do. That's good.
So any other questions for Lucia? If not, I want to thank you again, Lucia, for the wonderful presentation. Thank you very much. We're not in person, we cannot applaud the way we want, but you get the message, you get the idea.
Thank you so much, Stephen. We'll keep working.
All right, thanks.
Made a lot of progress. Fantastic.
Thank you for the feedback and the nice questions. And and even Gil, your question was a hard one and very thought-provoking, but I think we need to keep that in mind and see how is it that we're moving forward with these applications in different areas in dentistry, right?
Yes, definitely.
I'm looking forward, maybe 2 generations or something. Takes a long view.
Okay, Gil, you and I are long going to be gone before these 2 generations. Maybe Josh will still be here, but for sure Brian and Kevin will be too. Maybe Josh and Reza will be here.
Come on, we're not going anywhere, Lucia. Forget that talk. And Gil's going to be around for another 20 years, man. Come on.
Oh, why not?
We're going to the longitudinal aging seminar next.
Come on.
All right.
Okay, thank you so much.
Thank you very much.
Thanks, everybody. Thanks, Josh, for saving it all together. Bye-bye, Lucy.